And we're back, folks. So did a brief little stop up top. Plugged our hose back in, wiggled some cameras around, and we're back in the water for about 2 hours. So a little less for stream. But we are going to be sticking right around 200 meters, maybe get a little shallower. We've got our 2 detritus samplers and the 8 suction buckets to fill. So let's see what we can find. I'm going to bring up mission control audio for y'all on this, our second final dive of the expedition.
Final dive. Hey everybody, this is Karen Osborne from Smithsonian National Museum of Natural History. We are wrapping up this fantastic 2 weeks of midwater dives off the coast of Brazil. We've been about 360 miles off the coast of Brazil for the last 14 days and diving the ROV every day and finding amazing things. So this is our last chance. We're keeping it pretty shallow. Right now we're at 200 meters. Oh, it's caught in the sled. We're just trying to get the vehicle set up here. We are out here to look at all the amazing midwater diversity. There is a phonema going by in the zip cam. So we have a bunch of different ways to image animals, not just the incredible 4K camera that we have that gives us incredible detail on the morphology of animals.
Thank you. All right.
But we also have the 2 pieces of equipment that you see in the picture now. On the left is Iris. This is a planotropic lens on a camera system with red lights that allows us to get the 3D structure of animals, allows us to measure things in the midwater, which is a really challenging thing to do. And then we have deep PIV, which produces a laser sheet. We pass animals slowly and steadily through that laser sheet to basically give them a CT scan so that we can see their inner structure. Both of these pieces of equipment give us different types of information about the animals, but both give us 3D type information. One works really well on opaque animals and one works really well on transparent animals. I have to say that DPIV is one of my favorite pieces of equipment because it gives us 3D structures of mucus filter houses. It gives us feeding structures. Other animals make mucus feeding structures, mucus egg cases. There is a bunch of organisms that, single-celled organisms that have mucus structures around them. And all of these things are something we cannot collect and view on the ship because they're incredibly delicate and they get damaged when we try to collect them. So we really have to take this equipment down and image them in situ, undisturbed. And this equipment lets us do that. So it's really exciting. We are having a little bit of issues with the camera, so if it blacks out for a sec, it is a connectivity issue. We decided not to fix it because this is our last dive and we wanted to get down here and get as much as we could. Yeah, it's like super gainy or something on this. There are a lot more amphipods up here now. We stopped and looked at 200 on our way up from the last dive. And we didn't see too many now that it is nighttime. Everything is coming up with its diel migration, coming up to the surface to feed on these productive, well, a little bit more productive waters where everything has sunlight. But under the cover of darkness, they can come up here and hunt and feed.
What are you feeling?
It's still like has this like green shimmer thing going on that we didn't have before. Is that a connectivity issue or is that just a setting issue? Maybe it's just my imagination. That looks fine.
Does that look better?
Yeah, I think so. It's a little hard to pick out the specs. What lights are on? Do we have
We have
different
all
lighting than
the
we
lights
usually have?
except for the lowers, and
Oh.
they're set to the 70.
Okay.
Only differences is right now the ISO is at 160 and not 500.
Okay.
Actually, it's at 500, which would make it
Really bright.
really bright.
Yeah. Okay.
It's actually green.
I don't know what's going on. We could brighten it up just a bit.
Right there. That's it.
Yeah, just a
It's,
touch.
ISO, maybe a little more.
It's really strange.
Filter in again?
No, we did not power cycle
We
it.
did the power cycle.
No, but we can. Did that?
Let's try it.
Okay. Alright, I'm gonna hold station. Here. Alright.
Can we turn the
Put it on.
suction on pretty strong?
Yes.
We'll just pretty much the whole dive, whatever bucket we're on. Love it.
Out of it.
Mm-hmm. All that air in the hose.
Alright, so home station.
Plenty of Amphipod.
Coming
There's a
down.
white house going by with the sit cam. We're fixing some of the settings on the camera, or attempting to. So bear with us a minute. So this has been a really fun and exciting expedition. We've had experts out here from 13 different institutions around the world. We have University of Western Australia, Tohoku University, JAMS Tech, both of those are in Japan. Federal University Rio Grande here in Brazil, University of São Paulo here in Brazil, University of Alaska Fairbanks, Stanford University, Harvard University, University of Rhode Island, Smithsonian, who am I forgetting? University of South Florida, St. Petersburg. We've had this really Bigelow Labs in Maine, and we have really enjoyed working together. We had a few challenges with getting, all our equipment here and things, and everybody has really pulled together to build equipment that we needed and help each other out. And it's been just, I think one of the favorite things about this exhibit or expedition has been the team and working together to do the work we plan to do. So I'm really proud of everyone, and it has been really exciting to learn about all the different groups. So we have experts here on engineering, right, for the equipment that we've built, the DPIV and IRIS. We have a shadowgraph, which you can't see. So when we suck an animal through the suction sampler, before it makes it into the collection bucket at the end, it goes through a shadowgraph, which gives us a nice view of transparent animals. We can see all of the structure in super fine detail, much more fine detail than we can on the other imaging. We also have a camera we can't see the feed from until we get it back on board, and it is, we call it the lunchbox camera, but it's actually a stereo camera that gives us a much wider angle view of what's out there from when we see those big animals, and to have a better idea of what kind of documenting what's in the water column as we come and go. So we have a bunch of engineers who've designed and built all of these equipment. We have a bunch of taxonomic experts and experts on imaging. And we have two systems that we've, sort of two systems, multiple systems actually, 3 different systems, 5 systems that we've built to study the animals in the lab, to study their behavior, to document their morphology. We've been able to look at microscopic animals and actually watch their cytoplasm or their living tissue flow around on single-celled animals. document fine structure of the eyes of some of these amphipods you'll see us collect here in a few minutes. It has just been an incredible week. Wow. Beautiful little siphonophore just drifted past us. So it has been an incredible week of looking at incredible animals, showing it to you guys out there what we see, and yeah, learning a lot of new things. So it's a little bit sad that this is our last dive, but we hope that we will be back to do more and that more people will use SuBastian to do midwater work. Whoa. That is very bright. Trying to get the camera set up. What is this guy here? That looks like Doliopsis, possibly.
There we go.
It's just off to the right.
Nose down.
There it is. It's just back in view. It's an interesting shape. Can't quite tell what it is yet, but since it doesn't immediately tell us what it is, we're going to take a closer look.
And centering.
It's a rhizarian.
It's a rhizarian.
It's called a cell.
No.
In the house of Jack. No, it's a chain of Doliopsis.
It's a chain of Doliopsis, and the red is the dinoflagellates. How come one of them is so much bigger, Russ?
You zoom in, that's the pigmented
Look
version of the Doliopsis.
We could
That's some
could
nice
do
ring
lowers
to it.
on and
Oh, it's
there's
huge.
mid-
Is that the
Almost like a
adult?
starfish.
That's
It
the
seems
adult
to
with all the
be
babies.
larger
Holy
form. This one's
mackerel.
got all smaller versions of the chain.
That's the slow one, is it? Coming off the.
yeah, and then the capex, that top left
Yeah,
one off.
that's
I
our
was down at
Frosina.
Frosina.
Yep.
Yeah.
It's the first time I've seen such a kind of mixed range of sizes.
Yeah, I usually think of them as all the same on a chain.
Man, they're so high compared to their length, aren't they? Are these the ones that have the rotator polyps on the side?
Usually the pigmented ones do have a polyp. There's a polyp right there, maybe.
Do you want to collect this, Russ?
Do you want to collect this, Russ?
Want to collect them?
Well, we've got lots of these.
So that's a no.
Ah, the streets,
There's
yeah.
a Streetsia.
This Doliolula.
So we are sucking pretty hard, right?
Yep.
Okay.
Let's go.
So we're running the suction
I don't see
sampler
anything in here
continuously
telling
up
me
at this depth
there's
because we
anything
want to
different
catch
than
some of
the
these
other
amphipods, like this long skinny
Sorry,
one
just
that
so
just
you
ran
know, it's
through. This is Streetsia, and Streetsia is really interesting. Do you want to look at this further, Russ? And if so, what details do you want to see?
Russ, do you want it to look at this more, or are you done?
It's Doliolula. It's a nice
do
example
you
of
want-
the animal with the stolon attached,
Yeah.
showing some big differences. The large ones like this tend to be surprisingly pigmented with this
Yeah.
orange, and we don't see it on smaller specimens.
Is that pigment the dinoflagellates, or is that just pigment?
It
Can we-
looks like a chromophore. I mean, they're
It looks
regularly
like a
arranged.
Big enough.
chromophore.
Yeah,
Okay.
they're regularly arranged, always in the
Haven't
same
we scanned
place.
these, or probably
Um,
not very
No,
much?
something this big.
a
I think.
bigger one like
Can
this
we-
probably has the hydroid on the
Can we get pivot,
current
please?
cycling.
Yeah.
A little white on the end that's at the top.
EPIV.
I don't know if you'll be able to focus in on that to see it or not.
Do you guys want to scan this?
Yeah, I don't think we have a scan of an
Okay.
None
one
of
of
the
these.
one with this kind of size differences.
not the size. Yeah.
And the larger one is probably at the larger end of what we collected.
Yeah.
Yeah.
Looks big, man.
Okay. Let's DPIV it. Laser is on. There's an Alciopid swimming by. There's Streetzia back there. These little amphipods with the arms sticking out the
looks
side
like
are
it, huh?
Fracina.
But it's not, right?
Fracina has two pairs of
have an
eyes.
example of where
And these really cool,
I was
big,
right and,
really modified
know.
legs
and
that
Dougal was in.
they actually use as hydrofoils, which I just think are so cool. We always thought these big, spiny, toothed arms were for catching prey. But when we watch them swimming around out here, we see that they actually use those arms as hydrofoils to help them in their maneuverability when they swim. So that's the kind of observation we can't really make in the lab, because in the lab we can't give them enough space to swim the way they would naturally. And so we wouldn't necessarily have noticed that before. These long skinny guys you see going by are Streetzia. Streetzia has a cylindrical eye, and the length of that body, they have a rostrum that sticks out the front because they can't actually see in front of themselves. They can only see in a single stripe around their bodies. They're basically line scanning the environment around them, so they have to swim to build up a picture of what's near them. So what we're doing here with DPIV, it's a laser sheet. We have a camera looking at it. We turn off most of the other lights to reduce the contrast of all the other amazing animals and particles in the water. And we try to drive at a steady, even pace, drive the animal through that light sheet so that we get this very nice slices of image slices of the animal as we go through. I don't know how you can see it in the dark like that. There it comes. That is a Phronema. That was a very nice scan of a Phronema.
Yeah, let's try the camera then.
The lights, yeah.
Where did they go?
It's a salp, right?
I would put Phronema. The salp is gone. Just the tunic left, highly modified by the Phronema that lives inside. Phronema is another amphipod. They catch salps or pyrosomes or sometimes calicophrans, and they take the gelatin from that animal and reshape it into a barrel-shaped house. It has a specific diameter at one end, a different diameter at the other end, and they then carry it around. They use it for buoyancy, they use it for protection, they use it to house their little, whoa.
What is happening?
They use it to house their young, the females. I don't know, but that suction bucket is going to be full of goodies. So many goodies. Slowly, so we don't blow these guys away. It'd be nice to just let a bunch of them get in there. Maybe try to catch one of these big guys with the legs sticking out the side. Yeah, they're the kind of really fat ones, and if you look at them, they have these look like little shields sticking out the side. So not that. They're also the slower of them. They're not slow. They're slower. So I think if we hang here for a second, they'll come back. They are attracted to the lights. They expend a lot of energy building pretty incredible eyes, but despite spending all that energy and resources building these eyes, really large eyes. Okay, there's one.
One's
Yeah.
coming up.
They actually can't see all that well, but they focus all their energy to pick out just the information that they need. Because it's very difficult habitat to see anything in. You have to have super sensitive eyes to be able to see in the dark. Interestingly, most deep sea animals, their eyes, they're not used to being able to see seeing these really bright lights, and so their eyes oftentimes take a long time to recover from. There's one, just this guy. Just video of how they're swimming is really useful, even. Whoa, go! Thought he was gonna go in. But hyperids, interestingly, their eyes recover incredibly quickly from extremely bright light. So that would suggest that maybe they do migrate, not just in the dark, but that they can handle fairly bright light. Many hyperids, none of the ones that we're seeing here, but many hyperids are thought to be associated with gelatinous zooplankton. And so the thought is, if they're sitting on the gelatinous zooplankton, there's one.
A couple
Yep,
things.
yep.
Yeah.
I mean, there will be a bunch of them in there, but these big fat ones seem to get away. They seem like they would be so slow. They're really not aerodynamic, you know? And yet they are really quick and really maneuverable. Part of that maneuverability comes from their appendages that they use to fly. What is all of that stuff every time we...
Ooh, ooh.
Like, I don't think it's individual. It seems like bubbles. Is that hydraulic fluid? I hope not. Good.
I feel like it's a bunch of scales.
Okay. Yeah, I think not quite as glittery as I think, but that's bubbles. Oh. And also the EK80.
Let's see.
It's a lot of streetsia out here. We haven't been collecting very many, but we certainly have been seeing a few. There's some, see in that hole? There's bubbles coming up in that little chamber there,
Yeah.
like there.
Yeah. Yeah.
Right here, almost like something. Is that mineral oil company?
Message Sunny. Yeah.
Coming out of the arm.
So,
That's not good. We've got mineral oil leaking
Yeah,
out
we gotta
of
go.
something. That's it. Can we switch buckets and leave the suction on super hard?
yes.
All right. We are pulling
Index
an hour and 15 minutes early because we have a leak.
Oh.
We have had 19 fabulous dives. We have collected some animals that have
ROV.
not been seen before.
We
We've
are
collected
gonna have
multiple
to
new
recover.
species.
Okay. I will just come to a stop then if that's okay.
Sounds good. Roger.
So if we're recovering now, will we come back in or is that game over?
Game over.
Game over. Yeah.
Okay, I'll copy.
Okay, I'll tell the exhaust to that arm, so should be alright.
Here.
How much did it go down by? Do you put it on there?
Yeah.
Hey, is there any specific reason why, just so I can let people know?
One of the arms have we have a little leak.
Okay, I'll copy, thanks.
We have 40%, it shouldn't go down anymore.
Looks like your port arm's going too.
Yes.
Yeah, look,
That's probably from the,
because they're tied in together, but look.
the main pump. Yeah, the main pump is frigid.
Yeah.
Go forward.
Forward.
Well, we have stopped now. The leak.
Mhm.
We come on deck now, already doing it.
Yeah.
So you could probably continue the dive just
Sonny.
Thank you, Sonny.
Thanks, honey.
Okay, never mind. We're gonna keep diving.
Bridge, RV.
Yeah, go ahead.
Alright, cancel that. Yeah, we shut down that arm. It seems to be holding, so we're gonna try and finish the dive.
Okay, okay.
Not done yet, kids. Not done yet.
Alrighty. We have
This has been wonderful.
an hour and 13 minutes.
Alright, dive. Back to 200. Back to
No,
200.
you don't have to actually. We can work
Take it
between
easy.
here and 150. We'll just hang out slow, let the amphipods come back, keep sectioning them up, find some cool larvaceans to stick in those D
You want like
samplers.
50% suction or?
Oh, I thought you said you
full?
want like 50 larvaceans. Yeah, like 50 larvaceans would be great.
Okay. Sounds good. So 100% suction then. You tell
Like
me.
70.
70? Oh.
Yeah. What do you usually put it on when you're like really trying to catch a fast animal?
Oh, 100%.
That. You can put it on 100%. These guys are pretty wily.
Let's see. All right, so we'll slowly creep up and.
Good.
With our, yeah.
Good.
With our, yeah. We're gonna try. See if we can sink this basket.
Oh, there's a twofer. Whichever one is lined up better.
Oh, he went right to the
Ah.
outside. You can save it. We
It's
can
amazing
save it.
how they really look like they're in the tube
You want
oftentimes.
to bring that out for you?
What?
So, suction also?
Would it be
He's-
helpful to
he's
have
going
the
now.
back of the section note? See it? It wasn't going in.
But
Although then you
I'm
wouldn't be able to
looking-
see it where
yeah,
when it went
from the
back there.
other-
There's a
okay, we
bubble.
still have that little bit, but we're still at 43.
That was a
And the
bubble.
rest.
Yeah.
So,
Oh, streets, yeah. Come on, buddy. Go in.
Which one? Which one?
The red one that was swooping around in our camera. He was doing loop-de-loops. He would be very difficult to catch. But maybe they'll just go in. We got 4 the other night.
Yeah, that was.
They are fast,
Okay.
which is incredible. They can push that big old body around in the water with 3 little tiny paddling legs. Well, 6 little paddling legs. 3 pairs. Oh,
That's out. I was just making sure we were racked out all the way.
okay.
Do you want to look ahead or? Look
Yeah.
at the,
Yeah, if we can.
oh.
Maybe just if you put the nozzle out, we can still see it and see what's going in. Because you know they're all going to swim in there.
There
Feels
you
like it's a little bright up in the top right. So many amphipods. Oh. Oh no, I think that's just an amphipod. What about that? It's
Oh.
there, yeah. Oh, there's an Alciopid. I think we're looking.
What are we looking for? Which one?
It
You
was
wanna
some
focus on?
Medusa-like thing. I don't see it again,
Oh, I think it's right. Where was it? Right up top
but that amphipod is just dying to go in. Come on, buddy. Yeah, that was an Alciopid. There was 2 Alciopids came through. Can we zoom on this one?
Okay, all right. And,
Platycencellus. Ostreetzia. Look at them go. I really want to capture what they're doing with those legs. They seem to be using them as hydrofoils. Just kind of crazy.
Just some cameras or lights if you'd like.
Yeah, that would be great. Or iris down, maybe. Just... Oh, you crazy little swimmer.
Paper planes. That was really good.
So these are hyperid amphipod. Amphipods are a group of crustaceans, like the little beach hoppers you may have seen hopping in front of you when you walk on the beach and there's a lot of seaweed. You also find them in tide pools, in among the algae and stuff. Amphipods, there's about 10,000 species of amphipods, and there's about, this group called hyperid amphipods are about 280 described species, although there's probably at least twice that many actual species out there. They just haven't been described yet. This one here is a really good example. This is supposedly Fracina semilunata, but every single Fracina collected all over the world is automatically identified as Fracina semilunata. And they probably aren't. So one of the things that we are doing as part of this cruise is collecting them from this location. We're sequencing them so that we can tell if they're different. If they are different, then we will look really carefully at the morphology and figure out how to tell them apart. There's, oh, look at that. We can look at that little fish.
Oh, look at you.
He's right up, looking at our sit cam. So shiny.
Oh.
So very shiny. Hello.
Ooh. Ooh.
Whoa.
And he's fixed. He's gone. Try and follow it if you want, or just do our own thing. He's out front there.
Yeah.
This is a different one. I think that's it.
I think that's the same one.
Yeah.
Maybe.
He is not the most efficient looking swimmer.
Ooh, shake
Oh,
it off.
little shiver. Shake it off. I can do this. I can get away.
Yeah. We do our lowers and uppers off and all the bells and whistles, please.
Iris down a bit.
Follow you up, buddy. You may have gone faster than I can at the moment. Yep. He's gone. Alright. Onward. He's gone.
What? Are you saying my amphipods ate all the jellies? I mean, maybe. There's a little jelly they missed. There was an amphipod on it, though, I believe.
Have you been getting many of the amphipods in the nets?
No, not nearly as many as one would expect, considering that we're towing through this water.
Yeah.
When they're up here, somehow they are escaping. We are getting some, just not nearly as many as we get when we just turn on the suction and go for it, try to collect an animal.
Now, obviously, folks watching the dives don't get to see the net tows. What sort of stuff have y'all brought up?
Oh, we've been getting some really wonderful specimens, especially this morning. The very last tow, we got an anglerfish in the tow. We've been getting a really nice assortment of pteropods, which are mollusks, some of which have shells. Those are the thecosomes, or euthecosomes. We've been getting some gymnosomes, which are the sea angels that are related. They don't have shells at all. They eat the euthecosomes, generally. We've been getting some pseudothecosomes, which means they have a gelatinous shell or a very, very thin chitinous shell. So the pteropods are always one of my favorite groups to see. This morning we got about 4 different kinds of glass squid, very small ones, a couple other little squids in there, lots of different kinds of shrimps and mysids and euphausids. We got a nice selection of ostracods today, which look like little clamshells, but there's a crustacean that lives inside of them.
Seed shrimp, right?
Yeah, with really bright red, really large ostracods. this morning. Those were nice to see. There's a nice sparkly.
Much in the way of bioluminescent organisms coming up, or
Yeah, probably. We have a lot of jellies in the top. What is that? Oh, it's a little larvacean house. Yeah, a lot of jellies. We've been getting a lot of calicopherans. Those are still very happily swimming around. We call them rocket ships. They look like a little rocket and they pulse. What else have we gotten? We've gotten a lot of fish. This morning there was quite a variety of fish in there. Of course, lots of cyclothony, which are bristlemouths. Oh. There's a house. Oh. Hit the brakes. It is very tricky to fly in the midwater because the vehicle is pushing along a bunch of water with it as we drive. And so when you stop the vehicle, all that water keeps going ahead of you and blows the animals away.
Zoom in from a ways as it gets a little further.
It's a funny little house, Russ.
Very slowly bump in. Yeah. Yes, please. That would be. I'm gonna try and just hold it here, but stay far enough away that I'm not.
So this is a larvacean house or a
Right.
feeding filter.
Those
They
actually-
produce these, basically sneeze them out and then pump them up. They have a very specific filter size. They're trying to get particular size particles with these mucus filtering structures. What we're doing is looking for the animal in the house. They look like a little tadpole. You usually see a head, and then you see a long tail that pumps in a kind of sinusoidal pattern. I don't see anybody home in this house, so we can leave it.
Pretty small.
It's very small.
Yeah.
I'm not. I think it would be too small to scan.
Right.
So the animal pumps the water through these filters and concentrates the size particles that it wants on a little final filter level. And then when a final filter is kind of full, it grabs the edge of the filter and flicks it so that all the particles pop up and it sucks them in. They are super cool little animals, but incredibly delicate. How they make these incredibly specifically structured mucus houses is just, I think, one of the most fantastic things. And
Still maintain.
DPIV is one of the ways we can see what the structure of these houses is like, because it's not something that they build in the lab. I think the animal is there, just there in the center. I don't see it pumping its tail, but it does. It's just sitting there.
Don't go down.
I think he's there. Oh no, you can see him pumping. He's pumping. Do you think, how big is this?
Yeah.
It is. Oop, let's
Can we,
push it up. Oop. Oh,
can
that
we
side
put it
cam
in
there?
oh, we could try to put that in a D.
Yeah, so I
Let's try that.
Okay, we're gonna try to put this in a detritus sampler. That is the gentlest way to collect something in the midwater. Basically, we fly this giant vehicle up to the specimen
We're gonna
that
try
we
and
want.
sample it?
Yep,
Is that- Okay.
D sampler.
D sampler?
which side?
Yeah,
The
either one.
shoulder.
We fly this giant vehicle up to the organism and we fly it into this canister and then close the top and the bottom.
Alright, portion out.
Basically, you can collect the organisms without them even knowing that they've been collected
We're
because
gonna try and
they
de-sample this.
can be undisturbed.
Up and down.
It's ideal for these really delicate
Go for
animals.
the inner one, inside number 4.
Bumping down a suction hose is not necessarily the best for some of these delicate animals, so
Cut off automatically.
Maybe. I don't know what's the for okay, but how's what I'm guessing.
when we get up close, we'll have to try to see if it's still in there. I don't think it would have left since it is perfectly undisturbed.
And we'll test flies.
Slowly. Oop. Alright.
SD sampler is ready to go.
Okay. The lights are like, it's so blown out in the science camera.
Yeah,
I can't.
it is.
It's
Can we
horrible.
iris that down a bit?
Uh,
Although then it maybe that's better.
Better there.
Yeah, that's much better.
Thank you.
A little bit of a billow.
Harvard.
There's an amphipod in there.
Uh,
One of the other species of hyperids that we're seeing here is these little round guys. Their name is Platycellus. They have 2 pairs of eyes. They can roll up in a ball. They have a little latch on the sides of some of their front legs, that the back legs and the tail fit into to lock themselves together. I think the animal is still there. There's still a bright spot where it was. Streets here just went by. It looked like it had some kind of a parasite dragging on it, which is pretty unusual. All these Phracina. Nice and gentle.
Where is grass? See, there he is. Okay, coming up. Over. Oh. He's going. He's moving.
Got places to be.
Yeah.
Out front, low, and mostly invisible.
Oh my gosh. Yeah. This is the hardest question I've had to make so far. Okay.
It is probably the most delicate animal. I still see his tail beating in there though, so you haven't disturbed him enough to make him ditch his house.
I'm trying to see him.
Well, so we get partial signal through.
Under? Oh, no.
Nope. Oh, no, he's inside. Wow, I really thought he was outside.
Oh no. He just got hit.
Yeah. He would have failed. All right. Good try. Those are super difficult. I don't think you can find a more difficult animal to collect. It's not that
Just
they're
leave
super evasive, they're just so delicate. You have to,
Might leave the porch in next time because then I can actually just see straight.
yeah, that would help. And then you can tell where he's
That's
at.
horrible.
I wouldn't call it horrible. Don't worry. Oh, what's that? Oh, yeah. I mean, not that that's disappointing, but I'm really hoping for a pig butt, and that shot right down the barrel kind of looked like that. If you
Yeah,
didn't
yeah.
see the legs sticking out the side, but
Yeah.
I could suck
What's the
him up.
thing you saw this cruise that most surprised you, I guess?
Oh, the thing that most surprised me.
Or that you were most excited to see? Not that it surprised you necessarily, but either one.
So one of the new species really surprised me. Tomopterus or gossamer worms are one of my favorite animals, and what a bumpy house this guy has. Has not finished remodeling much. So we seem to have a lot of luck catching other things when we're trying to catch these guys. So let's try to catch this guy.
Try and catch it?
Anything, really.
Oh.
Any of these hyperids.
Anything.
Yeah.
Okay.
We seem to get the most when we actually spend some time just trying to collect other things. Can we iris open just a touch?
Yeah.
So the gossamer worms, so we just finished writing a paper describing 2 new species of gossamer worms. And so we, looked at all the characters, compared them to everything that was known. They have a really interesting tail that's different than all the other Timopterus, different than anything that's been described before. And so we felt really confident that we could call them two new species. They look similar to each other. One has a dark red gut, one has a light orange gut. They have different numbers of segments and several differences between them. Down here, we've seen two things that look exactly like those species, but one of them is different. It has a different number of segments. It has a couple of different characters. Its nose is shaped differently. Instead of a simple, clear, big, round ring of cilia, it has a double ring of cilia, and it's much smaller and raised. So there's some significant morphological differences. So it was really surprising to me to find another species very closely related to the one we just described. It also means we can be very confident that it's a new species. And since we haven't quite yet submitted that paper, we can quickly add the description of this new species based on the photographs and the video and the specimen that we have, a couple of specimens that we have. We can add that description to the paper and get it named within just a few months of finding it, which is really exciting.
That's very cool.
I think that really surprised me to find similar, closely related species down
Nice.
here. Another thing that's a little bit exciting or a little bit surprising is these swarms of hyperids. Hyperid amphipods are thought to be symbionts on gelatinous zooplankton, living on jellies and ctenophores, and lots of different other larger, slower moving zooplankton. Many of them are. They have, oh, are those bubbles again? I mean, I shouldn't point them out, but I should.
There are probably some that have
Chopped up
covered
underneath.
under.
Okay, good.
Yeah.
I like that. I like that theory.
Because the comps are still.
Yeah, our
They're
comp
still
levels are
okay.
still
Okay,
good.
cool.
Yeah.
Good. Good.
Solid.
You're now using it.
So I've seen these before when I was using a manned submersible up off of Cape Verde. We saw swarms of these animals. The key seems to be to move the vehicle slow. The hyperids are attracted to the lights, and all of these different free-swimming varieties come probably to hunt or because they're naturally attracted to light in front of the vehicle. And we see these huge numbers of them. And interestingly, even though we are half a hemisphere away from Cape Verde down here, we see the same composition: Stritzia, Platycellus, Persina, Phronema, Euprinoids, Paraprinoe. It's really cool. And I love watching these Phrycina swim. They have this modified pair of legs that's, when they're swimming slowly, they stick out to the side, kind of like stabilizers. When they want to stop, they spread them out quickly. When they want to turn, they sometimes use them, stick one out to the side and twist it, so then they turn very quickly. They seem to be using those legs as airfoils instead of the predatory appendages that we assume they are, because they have big teeth all over them. Teeth and spines, which we assumed was to catch. I don't know. There's a little oik house, probably too small to do much with.
See if we can just look at
I
it.
think it's really close. Yeah.
So
See, it's like if we see it after it's already inside the nozzle, it is not going to be an easy one to look at. It's funny because we look at the nozzle the whole time and we catch a few, but we don't see them going in. There's also a ton, when we do these collections, there's another Alciopid worm coming on, wants to come up and have its picture taken. Oh, there's a fish over there. So there's also a ton of small hyperid amphipods that are here. When we suction like this to try to get some of the big ones, we get a bunch of little ones as well. Let's see who that guy is.
Going after
The
the
fish.
All
Yeah.
right.
Go after the fish.
Walk away.
Oh,
Walk
it
away
looks like
from
the
worm.
Barracuda again.
Oh, yeah.
I don't know if that's what they decided that was, but that was the name I heard people bandering about. Oh, the worm wants its day in the sun.
Going to
They're
the
so
fun
cool.
worm.
Yeah, that looks like the same type of fish, no?
Very bright.
Very bright. Nothing like a little mirroring in the deep sea. So mirroring is actually a really great camouflage from the side, from things that are looking at you from the side, because it just reflects back all the surroundings. So you automatically match your surroundings if it's mirrored, and there's a lot of the surroundings are pretty much the same everywhere. Mirroring makes a great camouflage. Another strategy for hiding out, see how this guy is oriented up. So that is either because he is migrating up, or it's because if he's oriented this way, he has a much smaller silhouette for hunters that are below. In the midwater, a lot of the predators are in the deep, looking up towards the light coming down from solar illumination, either the sun or the stars. And so they're looking at all this backlit silhouettes. So everything wants to make its silhouette as small as it can, either by orienting its body in a way like this. Oh, that's
Follow
not a
that.
little squid, is it?
Follow that.
Yeah. It's probably just a crustacean, but
Looks like a
it looked like a squid to me.
little dove down.
So many amphipods. Surely some of these will go in the bucket. There's the fish again. I think we can leave both of them. That's a nice head-on view of Platycellus. Platycellus are these little round hyperid amphipods. They are a little bit like little armored tanks. They have very tough exoskeletons. They've expanded parts of their legs to shield the rest of their legs. And the rest give them a little protective coating. When they feel threatened, they roll up in a ball and latch their tail in, effectively turning themselves into a little hard sphere. So if something has to be able to swallow them whole, they wouldn't be able to bite into them because they're pretty tough. All right, we can
All
leave
right.
this guy. Marcella hasn't come running in.
Okay.
Super excited about this thing. So
We don't have to have it.
we don't have to have it. And I'm pretty sure if we tried to catch it, it would decide that it had had enough of us and jet off.
That's 15. And then the IP address.
It's another Phronema in the barrel. That one has a bunch of larvae. You see the ring of bright colored material. That is 100 or so juveniles that the mother has laid on the tunicate. On the salve. And the mother keeps them oxygenated, moves them around, catches prey, and feeds them, puts them inside the barrel for the larvae to eat, or the juveniles to eat. So many, yeah. Surely some of them will go in. I don't really see any swirling around in the bucket, but they're probably all stuck to the screen from the suction. Alright. 44 minutes left. It's the countdown to the end of science collecting. We still will have a bunch of animals to work on, finish some of our experiments, documenting them in the lab, imaging them, taking tissue samples for genetics. One of the objectives of this cruise is to build the reference libraries for midwater animals. eDNA promises to make a quick way to tell what's out there and see how it's changing, because you can monitor it really quickly. The problem is if you don't have sequences for the animals that are out there and you know which sequences go with which animals, it's not really useful to use eDNA. And so what we need to do is build the reference libraries. Effectively, imagine trying to write a research paper and you went to the library and there's only a couple of books on the shelf. You wouldn't be able to write a very effective paper, especially if it didn't have the animals in it that you were looking for, or the references in it you were looking for. It's the same thing with the genetic reference libraries. So we're trying to sample really broadly, produce reference sequences for all of these animals. Thank you, Sarah.
You're welcome. Gotcha. And cruise.
No, they're swapped.
This I did not.
Can we
Auto depth just because we're very slow.
turn off the suction and maybe index? Just
No, I'm good. Yeah, we should be coming.
Right.
Soon.
Suction off. Index.
oh yeah, look at them all. Woohoo! We have a ton of them in there. That is awesome. And then we can turn the suction back on.
That was number 3. We started on 1.
Yeah, Hyperids in 8.
Yeah, 1, 8, and 7.
Okay, section's back on.
All right, let's find some larvaceans. What are we at? We're 158 meters deep. We have 42 minutes left.
Oh, we are staying until.
I hope so. We have to be on deck at 9:30, so we don't have to pull till 9, correct? Okay. Oh, it's so funny how they so often line up and look like they're going down the tube and then they drift off. It's like, ugh, killing me. More cephalopods. Cephalopods are so amazing to watch. I agree. Charismatic megafauna of the midwater, but you have to love them. So we're hoping to collect some larvaceans. There's a tiny one up to the right. We want to find some of the big ones. We have not seen very many of them on this expedition. We've seen a couple. We scanned a really nice big house one day. We've seen a few large sinkers, but we have not seen very many large ones. They're also incredibly challenging to collect because the animal is typically quite small, completely transparent. And so keeping an eye on them while they're pumping away in their house is one thing because you can see the house, but once they leave the house, they're very hard to track and collect. So I'm surprised we're not seeing, last time we were at this depth, we were seeing tons of siphonophores, Forscalia. Interestingly, that Forscalia we collected, inside the swimming parts where there's no stinging bits, there was all kinds of embedded juvenile Hyperids, Platycellus, these big round ones that you see out here. But down in the stomachs were lots of other Hyperids that had been eaten. So clearly they hide out in the parts where they're not going to get stung to grow up, and then at some point they have to get away without being eaten by the tentacles or captured by the tentacles. So to me that was really interesting to see, because this is a question we've wondered, because a lot of Hyperids hang out on siphonophores, but siphonophores eat small crustaceans, and so we were wondering if it's like a clownfish thing where they have some kind of immunity that keeps them from getting stung. But it's pretty obvious from that one that, at least to that siphonophore, nope, we're stopping. It was pretty obvious from that they are fairly indiscriminate.
Is that a siphonophore or is that
I don't know, looks like a
Wee little thing.
wee tiny worm, possibly. We got a small alceopid in one of the plankton toes. Just very transparent, you just see the eyes. We can go on, it's too small to
I bet.
do much with. What's this guy though? Oh, there's one. We don't need it though. Unless you want to just run it down the suction nozzle. I would take it. Actually, Renato would probably like it. We got one the other night and he did not get the quality video that he wanted of it swimming. So let's try.
Try to suck them up.
Yeah. Try to suck them up. We will want to cut suction as soon as it's in the nozzle because it's a little bit delicate, definitely more delicate than the Hyperids.
Do you want full suction, half suction?
I would say 70%. These guys are not super strong swimmers, but they will suddenly shrink up when they feel the suction probably, which hopefully will just make them easier to get in there. They're really beautiful when they swim. They, the entire body undulates, and then depending on how long they are, the entire body goes around in a spiral, in a helical pattern to get through the water. And on the front of that, you see two red dots on the front of this animal. Those are its eyes. It has single-lensed eyes, just like ours, and can see almost as well as ours, except obviously its eyes are much smaller. There's a little bit of ink up there. Must be a squid nearby. Still here. Oh, he's out of my view. There's a little bit of a salp chain. I lost him. Oh, up there. All right, he's coming in. This is a very small alciopid. Very transparent body. But they often have a little bit of pigment just at the base of their parapodia, which I think is what's giving it this little bit of a red tinge. A lot of the animals you see down here are red or have red on them. That is because red is effectively black. There is no red light down here. So if you want to cover your, come on, buddy. Yes. Yeah, that makes it very hard to triangulate where he's at. He's up here.
There, you can barely see it in that one now. I zoomed in a little bit. Just can't pace here.
He's here. Oh, sorry. Too excited with the finger pointer. Teeny tiny. Typically one of the eyes is about the same diameter as the body. There's several different genera and a bunch of different species. They grow indefinitely. They just keep adding segments. Sometimes they'll break and they regenerate. Some species can get over a meter long, but only about as big around as a pencil. Some of them get much thicker, but not quite as long. Some of them are super pigmented. They ink, which is interesting. Not exactly sure where that comes from, but they obviously have some kind of glands, I believe, on their bases of their parapodia. Oh, he is tiny. So tiny. He's going to be challenging. I think he's stuck up there. I think so. Let's find something else.
Well, no wonder I never saw him with the other camera.
Does make it incredibly difficult to catch them if they are not visible in at least two cameras. Great thing about this vehicle is they have two cameras looking crisscross across the front of the, like kind of wide angle cameras crisscross across the front of the vehicle. We have the main science cam that gives us really high resolution image. Then we have the special camera for the deep PIV on as well that gives us another view. And we try to use all of these different views to triangulate where the animals are when we're trying to collect them. There's another krill or shrimp. Oh, hello. That's also an alciopid. One of the ones with smaller eyes. Bigger bodies, smaller eyes. So tonight is the last night of mid-eats. Nothing like having 4 square meals a day. But for those of us who've been working around the clock, it is really nice to have that bit of sustenance in the middle of the night. Typically, we just eat popcorn all the time in the middle of the night when we get hungry. But here we have actual full-on meal, often one of the better ones of the day, which is pretty crazy considering all the meals are really good. But there's always something a little special on the evening. Are we looking to the side? It feels like it's fairly dark over here.
Dark over there, yeah?
Yeah. That's
ISO
good.
is back to 500.
I think it was just because we were looking off to the side. Good. So many amphipods.
Fishy coming along.
Go
Fishy or
fishy.
squid?
I think it's a fish coming into view here, chugging away. It's got really large fins on the side there. It's a bit unusual.
I think he's coming towards us.
Suicide fish.
Like,
Just gonna fly
like, I'll
right
show
over the
you.
top of us. How fast can you back up? I can go that direction really quickly. It's a little larvation house out here. Teeny tiny little one. It's an interesting little lower structure on it. Usually see those. Trying to find a nice big one we can stick in a D sampler. Last few minutes of collecting here on this Designing the Future 3 expedition. So there's a pretty tight layer up here, possibly just a little bit above us. Can we work our way up a little bit?
Sure.
What is that? It looks like it has, you think it's mung? Certainly shaped amorphous enough to be mung. Probably a collapsed larvation house sinking down. These are a really important source of carbon because once you aggregate all the mucus and the microbes that are living on it and all the particles that they have attached to it. Oh, it's a radiolarian. Uh-oh. Oh, it is. Rhizarian.
Oh, get the camera ready.
even that is really.
Rhizarian, I think we have a Rhizarian.
150, 157 meters. We have a rhizarian.
Blurry this is.
It's one I've never seen before.
We
but
have been getting
I
these
think we
in
only
the
have 8
toe.
canisters available, but I don't know how to do it.
You can suck it up.
Just another new species.
Just another new species.
Suck 'em up.
Okay, yeah, let's suck them up.
That's a Rhizarian. That is huge. Feels larger than Rhizarian. I think Manu must be sick.
Surely not. He doesn't do that.
Something's wrong. Does he?
He does every once in a while. So this is a colony of single-celled animals. There is a gelatinous sheath around the outside of them. They're photosynthetic, which is why they're up here in the shallow water. That's about all I know about them. All right, in the bucket. Well, it's not in the bucket yet. It's in the tube. Let's see. Can probably turn off the suction and just drive forward. Are we driving forward, or is there still suction
There's
on?
a little bit of suction.
A little bit of suction.
And
Okay.
driving forward.
Okay. And still no sign of him. We should have some really nice images of this in the shadow cam as it passes through the chamber that is imaging everything that goes through. Maybe just a little burst of suction. See if he comes through. He may have just come in and zoom right around.
There it is.
Oh, there it is.
Nobody said Tina.
All right.
That's awesome.
Nobody said Tina for.
Done. Onwards.
No,
All
I saw
right,
this. Nobody
let's go to
said
6.
You said Rhizarian.
That is Rhizarian, supposedly.
Oh, we did say Colidarian.
Yeah, let's just find stuff to suck.
Wait, which
Come up.
one? You guys gotta make up your mind. 'Cause we need to know what to write. Rhizarian, Colidarian?
It's definitely Rhizarian.
Rhizarian.
Need a tuna force view.
Rhizarian.
That's a good one.
I think it's a Colidarian.
It's not there.
Did you see? We have not actually seen any ctenophores. Not a one. Very few jellies. They're eating everything. Actually, when we worked one time in Oceanographer's Canyon, every single animal we shone the lights on, the misto would and just decimate it. Everything.
Is that too
It
dark?
was crazy.
No.
The misto is one of the types of high-paired amphipods, and at the museum we have a bucket from one toe that is a gallon bucket, and it is 2/3 full from one toe of the misto. So they get really abundant. They are a really important food source for fish out there. They also have really interesting eyes, which is why we're studying them. They also are a good example of midwater animals where we think we know the diversity that's out there, and then when we sequence them from different places, we actually find out that things that look very similar in one ocean and another ocean, or one location within an ocean and a different location within the ocean, even sometimes different individuals from the same location, there's a pyrosome, are actually different species. Typically, once we identify those differences with genetics, we can look specifically at the morphology and two houses. I'm not sure which one's bigger, but you can, they're both pretty small. Once we find that there are differences genetically between them, like major differences genetically between them, we look at the morphology and we typically find that indeed their morphology is different, and we can find a way to tell them apart as well. So there's probably at least twice as many species out there as are currently described. What is this guy?
Microscopic.
Tiny microscopic foramen. But it almost looked like it had a mucus net around it. Is that another Colidarian? Or is that a scythe? whatever it is, it's small. Looks a bit like ink. I think that is ink. Oh, there goes a fish again. Yeah, we can leave that. Squid ink, I believe. Squink, as we like to call it. Squid ink. They secrete, when they secrete the ink, they also often secrete fair amount of mucus along with it, which makes the ink stick together. Sometimes they can control the shape of the clouds that come out. They make them pseudomorphs or what?
So true.
It's all squirted out.
No.
Moving on to something
I was letting somebody else have the limelight for a second.
Can we open the iris just a touch? We have 20 minutes left. 22 to be precise.
What's your favorite squid?
They're gonna have to drag us out of here. I hear there's a lot of places to stow away on the ship, so
Big ship.
it's a big ship. Might be a little hard to convince the next group to do, you know, midwater, but
If I were you, I'd stow away in the others.
yeah, exactly. Exactly. Oh, what's that? It's kind of bright blue. Or maybe my eyes are just going a little wonky. Is it my imagination or was it very bright blue? I don't know. It blinked away. Now it's gone. It might be. I thought it was that spot, but now it doesn't look blue anymore. Maybe it was just a funny from the side thing. Nothing. A speck too small for us to see. Oh, it's very shiny though. Maybe it was. Maybe it's a sapphirina. We've been getting. Sapphirina is a little copepod. They're extremely flat, and they have guanine crystals in their exoskeleton, so that they are. If you look at them just from the right angle, they're really beautiful, bright, shiny blue. And if you look at them at a very slightly different angle, they are completely transparent. It's a very cool trick. Interesting thing to see when you're blue water diving and you see this glitter of blue beneath you, and then it's gone the minute you think about doing anything. That's just a Fracina. We're still suctioning, yeah? We can suction at like 100%.
Full section.
Full suction. I think we have 2 professional singers on board.
We do. Who's that?
I think so. Lakshmi is a singer. Julieta is a singer. I could do the dancing, but everyone would run if we did this, if I tried to sing. Trust me.
I was told that if I needed
for
like, to give
talking,
a speech
maybe.
that Joel would be.
I don't know. I can't really carry a tune. This is
We
the
might just have
problem.
to ask
And
him
I
to
just sing whatever I hear next to me. So like,
You should give
He'll
me a
love
birthday
it.
speech.
He'll love it.
I do love to sing. That is a potty salad.
We miss it by just the tiniest bit.
Copepod, ostracod. I don't know. It was pretty shiny though. There's another alciopid.
Wormy?
Yep. Little wormy. Look at these guys. They're just taunting me. They really are. Look at me. I can outrun you. I have these cool. Yeah. You know that at 8 58, we are going to find a Bathycordius finally.
Always. Always.
That is how these things work.
It's okay, we can give you the 901.
Who makes
Ooh,
the cutoff
I
time
love
for
it.
when we have to-
I love it. The captain in this case. We have to meet pilot boat in the morning in 2 days to take our spot in the harbor. So we need to stay on schedule. But, you know, we need that extra minute. He might give it to us. You know, they're just taunting us. They are really cool. I can't wait to see the footage from your chamber of these guys just behaving. Even just recording them with no
We're moving
stimulus.
because we got them as soon as they came in.
Yeah, that's awesome. I'm curious if Platycellus ever moved because they seem to just hang there, which is weird because they sink like stones when they don't move. I mean, like the little ones, the minute they stop swimming, they sink to the bottom of the pool.
Yeah, like every time I put them in, I'm always happy because I don't have to worry about
Something
getting the
there.
air water.
Yeah, right. They're just gonna sink. You're not gonna get squished out by the lid or anything.
No, they're pushing. There's so many
Don't
things
actually
about
know how
them.
big it
There's
is.
worms,
There are.
Tiny.
And it's downstairs in the water.
Of course.
Of course, it's tiny. No, it's just its legs. It's crazy. I think we can just put it straight in a D-sampler without looking.
You wanna try and do that?
Yeah, sure.
Okay, we're gonna try and desample.
Excellent. Do you
there we
want
go.
the porch out? Porch where it is?
Porch, porch out.
Porch out? Starboard. One.
Oh, such nice
No.
view of this animal. So you'll notice that this animal has a red gut. This is pretty common in the deep sea. If you eat something that bioluminesces, and 96% of the things out here bioluminesce. So if you eat something that bioluminesces, you don't really want your stomach glowing to alert all of your predators that you have been well fed. So they color their guts a deep red, and that covers the bioluminescence in their guts of the animals as they're being digested. Bioluminescence often in the deep sea is made by combining a substrate with an enzyme. Oh, there's a little Halicera probably. Just up. There it is. One shot. It is a house. There is a dot as if there's an animal in it still. Gonna have one shot at it. I do not see it in the sit cam. Ah, there it is. We're gonna try. We have also decided with the houses we had before that we put them in the eDNA, so we sequence the entire house and all the microbes and particles and everything that's on them, and it gives us a good idea of what is out there. Of course, only if there are reference libraries for the animals that are out here and the organisms that are out here. Oh, close. So close. Oh. This flying requires such fine control. Oh, no.
Yeah, I think we missed
It came out here, yeah.
Yeah.
It came out?
Yep.
Okay.
What's this guy? Yeah, you can open it up.
It was great timing though.
I know, it was perfect timing.
Great reflexes.
You
Yep.
said open it up?
Yeah, if you can
Okay.
open it. It definitely went over.
We didn't get the house right.
No.
Alright.
We did not.
Yeah, that was reflexes like.
Right.
Yeah, like we fucking. It was perfect. It was perfect timing. He just slid right over the top instead of going in. Looked like he was going in. Tricked us. I can't believe.
Get down deep for those green hills, or we're trying not to run drum.
We have 13 minutes left. What depth did you guys see them at?
They were probably more like 253.
Yeah. We are not going to be able to get there.
They might show up to. It's pretty barren here.
It is pretty barren here. Other than amphipods. Dougal says they ate everything. I don't believe him.
There's-
They are pretty ravenous though.
Yeah.
They usually leave some remnants. It is interesting that we mostly see Phronema in barrels here, whereas in Monterey we see a lot of free-swimming Phronema. Up in Cape Verde we saw a lot of free-swimming Phronema that didn't have a barrel. Seems like we should at least get a 4M or something from Manu. We did get a Colodarian. That was good. We got a worm. Also good. We've got a bucket, bunch of Hyperids in them. Suction is still on, correct?
Still on.
Oh. Not the tiniest house ever. A little teeny tiny ROV that can do all these little teeny tiny animals. Doesn't have such a big. Exactly. That can be done. Can we iris up just a tiny bit?
Oh, wrong way. My bad. Also, here we go.
We are 100% cooking in this corner.
Oh, there was nothing.
Go in the funnel. Yeah. It's better than a little space heater, that thing. Oh, there's a house down in the bottom right corner. They're so tiny, these houses.
I remember tomorrow.
Yeah, we really need to. I'd love to sign buy the copies of these guys.
Yeah,
Go through.
they're great. Let's have a look at this. It's probably just another Fracina, but it's moving a little bit different than some of the other ones. Oh, no, it's not. Take that back. Just fooling us. There's no jellies. There's no nothing. We can work our way up a bit. We have a hard ceiling at 100 meters. Right now we are at a hard 150 now. Okay. Okay, we have a hard ceiling of 150. So we don't have far to go up. But we are quickly running out of time. Oh, let's get that. That is some kind of a protist. There's also a little string of doliolids, but we'll take the protist. Suck them up, I think. Yep. Wow. Off to the right, maybe. There he is. This will make an interesting shadowgraph, this guy.
I'm gonna try sucking him up.
Yep.
Super close. I think it's super close.
There it is. Going up. That is a beautiful platycellus. Just wanted his time in the sun. Oh. We could take either one of those. The upper one is the one we were thinking of getting. This one. What is it, Manu?
Yeah, this is a 4 amp. I think it's most likely listed
I don't see any spines.
zoomed in on it yet.
There's no spines. It's a sphere.
It's just a sphere?
Just a sphere.
Then it could be a Allospheria.
Oh, there it goes.
Oh.
It's down below.
There's a copepod. I lost it.
I think it went down.
Well, there was two. Maybe we'll find another.
Yeah, if nothing was sticking out, then it's most likely Allospheria.
Can we iris open just a tiny bit? There's a question in the chat. Bioluminescent and these creatures are only reactive to the darkness in the depth. They bioluminesce in the depths because there isn't any other light down here, and it is a way to communicate. It's also a way to startle predators. It's a way to communicate with your conspecifics. There's a lot of different ways that bioluminescence is used. It is not stimulated by the dark. It is usually controlled by the animal. And we can stimulate it to see if we have a low light camera. We can stimulate it by bumping into them.
Sailfish.
In the lab, we can stimulate it by, just gently disturbing them, and you can see where it comes from, what color it is. Bioluminescence is a really cool phenomena and really interesting to look at. Some of the animals we've collected bioluminesce so brightly we can see it in a well-lit lab. I got a shrimp the other day in one of the toes, and it tail flipped in the bowl that we had it in, and you suddenly saw this beautiful bright blue cloud of bioluminescence.
Will there be a way to share, a way for Jamstack to share how the shadowgraphs are displayed? What do they look like? Nobody knows.
So the shadowgraph is a standalone currently, set up on the vehicle as a standalone, so we don't get to see the recordings from the shadowgraph until we get it up on board and download the images. Those images will become part of the record of this dive. There's an Alciopid up to the right. It was an Alciopid. So the shadowgraph images will. There's a nice shrimp down below. Big, long antenna. Actually, might be a mysid. The way the antenna were shaped. 3 minutes. And then we will sign off and hopefully be back sometime this year. Next year. Hopefully more midwater biologists will come out and look around different places. We've had a fabulous time exploring the midwater here off of Brazil with our Brazilian colleagues from University of São Paulo and Federal Universidad Rio Grande. They have been working on stable isotopes. They've been working on identifying, cataloging the fish that we've been getting in the toes. Coming to finish it off, Joel?
Yes. For closure.
The closer. Perfect. He says, I know how to deal with her. Just drive really fast up. She can't see anything. She stops saying, stop, stop, stop.
I'll be very shallow.
Yeah, no, it'll be quick. It will be quick. Oh, so many alciopids. It's at least 2 different species we've seen and collected so far. They're wonderful little worms. There's another one doing a nice helical swimming pattern up there. There's some really nice work looking at how salp chains swim, and that the longer the salp chain gets, it has automatically forms a helical pattern as it swims. We have 1 minute. 1 minute, Sonny.
Rules are the rules, huh?
No, we're just swapping out. You still
Just
have your
swapping. Yeah. Okay.
30 seconds,
Put
20
your hand
seconds.
on the joystick. I really was pretty sure Bathycordius was going to show up 1 minute ago,
10 seconds ago.
10 seconds ago, but it did not.
1 more minute. Oh. 10 seconds, 8.
10 seconds. 8, 7, 6, 5, 4, 3, 2, 1. End of designing the future 3. Ah.
Nice job, team.
Thanks, everybody. What an awesome expedition. All right. Signing off here. We are going to recover the vehicle, get our samples on board, do a few more experiments, watching how these animals behave, and head into port. Thanks, everybody. It's been fun.
Okay, bye.